Hello, my dear students. This is Indira. Today we are going to start with yet another topic. So, how are you, all of you? I hope you are following my videos regularly. And continuation of that topic, today we will do regulation of gene expression. And first we will start with what is regulation. I have given here a detailed points and notes. Each and every point I will be explaining, so that it will be very useful to you, and it will be like a notes also. You can copy down as the notes. It is very important topic for board exam as well as for the NEET exam. So let us now start with the topic, get into the topic. So what is regulation of gene expression? The meaning of regulation is controlling the gene expression. Now what is gene expression? A gene expression means a particular gene represents a character. This you know, all of you. A particular gene will represent a particular character. So this expression, external character or internal, gene expression can be both, either external or internal. So the character produced by a gene, that is called as a gene expression. Now this gene expression is also controlled. Regulation means controlled. All the genes are not functional all the time. So depending upon their requirement, a particular gene can be functional. So let us see some of the points. Every organism adapts itself to external surroundings. This is for every living organisms. And cells of organisms have the ability to turn the genes on and off. So depending upon where they live, their external surroundings, the gene can be functional or it may not be functional. And such control of gene expression help in production of specific protein whenever needed. So whenever that protein is needed, that specific protein will be produced. And how is it controlled? This gene regulation is controlled by various metabolic process, physiological and environmental conditions. So these conditions can control the gene expression. Now let us see. There are many examples where gene regulation is done both in prokaryotes as well as in eukaryotes. At different levels in prokaryotes and eukaryotes, the genes can be regulated. So what is the meaning of gene regulation is all the genes are not functional all the time. Depending upon their requirement, a particular gene can be functional. But all genes are present in all organisms, in all cells, right from the infant stage, right from very young stage. Suppose we take the examples of human beings. All the genes are present right from a child is born, right from the birth. But we know very well all the genes are not expressed at all the time. At the time of birth, all the genes are not expressed. Especially depending upon which protein or hormone has to be synthesized. That is synthesized only at a particular stage. That is called as a regulation of gene expression. Now in eukaryotes, at different levels we find gene regulation at 4 different levels. First level is transcriptional level. That means formation of primary transcript. What is a primary transcript? It is hnRNA. This we have done already in transcription process. Primary transcript means hnRNA. Now how is it regulated? How is hnRNA regulated? hnRNA has to be processed. So what is primary transcript? Primary transcript means it is hnRNA, heterogeneous nuclear RNA. This hnRNA is then converted into mRNA, a mature RNA. By capping, tailing and splicing, it is converted. So regulation means at any point, if any particular substance or hormone or any particular chemical is not available, then it cannot be controlled. It cannot be converted. That is called as a regulation. You know, capping is done, tailing is done and splicing. So at these three stages, if any of the chemicals are not available, then this conversion of hnRNA to mRNA cannot take place. That is the meaning of the regulation at transcriptional level and formation of primary transcript. Second one is processing level, that is regulation of splicing. So splicing also is regulated. Splicing, we discussed the mechanism by formation of spliceosomes. Suppose spliceosomes are not formed, then this regulation process cannot be taking place. Splicing cannot take place. That means there is a regulation, there is a control. Then transport of mRNA from nucleus to cytoplasm. This is another stage of controlling. So through nuclear pores, the transport must take place. Then there is a control at translation level also. Translation, we have discussed, translation takes place, there is a control. At cap there is a control and there is a tail formed. There are UTR, untranslated regions, which actually can help in efficient translation. If there is any change at this stage, then translation cannot take place. So at these different levels, it is regulated in case of eukaryotes. Now, let us now continue with regulation of gene expression in case of prokaryotes. The regulation of gene expression in prokaryotes. So before we get into the process, we will first define mainly two types of genes. First type are called as constitutive genes, or they are called as housekeeping genes. Constitutive or housekeeping genes are which are constantly expressed because their products are required for normal cellular activities. These genes are continuously they are expressed. Why they have to be expressed? Their products are required. For normal cellular activities. Like for example, genes for glycolysis and ATPs. Genes for glycolysis, always respiratory process has to take place. So always those enzymes have to be produced. Similarly, ATPase is involved in synthesis of ATP, energetic molecule, which also have to be synthesized continuously. So these are called as constitutive genes. Next is non-constitutive genes, or these are called luxury genes also, because these are not expressed all the time. These are not always expressed. They can be switched on and off depending upon the requirement of the cellular activity. Like for example, genes for nitrate reductase. Nitrate reduction process is taking place in plants. So whenever there is availability of nitrogen, then this enzyme will be synthesized. First, nitrates have to be available to the plant. Then nitrate reduction can take place and it is then converted into ammonia. So this process will take place only if nitrogen is present. Suppose nitrogen is not present, this enzyme cannot be synthesized. Lactose metabolism also in E. coli. Lactose metabolism. This is also all the time the gene cannot express. Only when lactose is available, the genes can express. Now, regulation of gene can be done in two ways. One is called induction, another is called repression. So let us see what are these and what are the examples. So induction. Induction is a process where addition of substrate will stimulate or induce synthesis of enzyme required for its breakdown. Suppose any substance is added. That will induce synthesis of its own breakdown. That is called as induction. And the substance which is added is called inducer. Like for example, in Lac Operon, this we are going to do in Lac Operon. That is called by induction process. Then second type is called by repression. So repression is the end product inhibition. When end product accumulates or its addition will stop its formation. Suppose some X is the end product. X is the end product. Say A is getting converted to say X. And if X is the end product, you suppose either X is added or if it is accumulated. Added or accumulated. Then it will stop its own formation. The X cannot be formed if it is getting accumulated. That is called as a feedback repression. This is here we will discuss the example of tryptophan operon. We will discuss the example of tryptophan operon. That is the example for the repression. Gene regulation by repression. So it can take place either by induction or by repression. Okay, now let us start with what is a Lac Operon and what is the meaning. So first of all, Operon. Let us see what is Operon. The Operon hypothesis was proposed by two scientists, Jacob and Monod, Francois Jacob and Jacques Monod. They explained what is an Operon hypothesis. An operon is a cluster of genes. Actually, it is an operon means a cluster of genes. It is a polycistronic gene which is regulated by a common promoter and regulatory gene. So, in an operon, there are some cluster of genes which will control a polycistronic gene. What is a polycistronic gene? A gene which can synthesize more than one protein, but all function the same. All the proteins will perform the same function. So that is called as a polycistronic genes. And operons are common in bacteria, in E. coli, and it is most commonly studied, and it is common in bacteria. Like for example, there is a Lac Operon for lactose metabolism, tryptophan operon for tryptophan synthesis, arabinose synthesis, arabinose operon, histidine synthesis will take place. Histidine operon, HIS means histidine operon, valine operon, valine. So wherever these amino acids are present, it is for their synthesis. Arabinose is a sugar for its breakdown. So breakdown of arabinose, synthesis of other amino acids like tryptophan, histidine, valine. So these are controlled by a cluster of genes, this metabolic process, and these cluster of genes together is called operon. It is derived from operation, operator and regulator system. Operator, regulator system. So based on operator and regulator, it is called as a operon, and the concept, the hypothesis is called operon hypothesis. So now let us see what is the general structure of an operon. So operon will have a promoter gene, inhibitor, or it is a regulatory gene. There is an operator gene. The two promoters, we call it as a P1 and P2. First promoter is for inhibitor gene. Second promoter is for operator gene. So promoter, operator. Then these genes are called structural genes. There are three structural genes in Lac Operon: Z, Y and A. Three structural genes. So all these together, all genes together will form a cluster called as operon. So now let us see one by one what is the function of these genes, and then we will go into the mechanism of the Lac Operon. So first let us see the structural genes. What are the structural genes? There are three structural genes as we have listed, Z, Y and A, and these are transcribed together to form a polycistronic mRNA. So what is the meaning we discussed for polycistronic mRNA? An mRNA which can produce more than one protein. That is called as a polycistronic mRNA. So first let us see what is a Z gene. Z gene. Length, this is length given. Z gene has a length of 3075 base pairs. What is its function? It synthesizes the enzyme called beta-galactosidase. This enzyme will break down lactose into glucose and galactose. This enzyme is useful for breaking down of lactose into glucose and galactose. Lactose is a disaccharide. It breaks down into 2 monosaccharides. Then the other feature of this gene, it also is involved in isomerization of lactose to allolactose. That is conversion of lactose to allolactose. That is another function of beta galactosidase gene. So what are its functions? Beta galactosidase gene produced by the Z gene. The enzyme is produced by Z gene. The gene is about 3075 base pairs in length. The enzyme will break down lactose into glucose and galactose, and it is also involved in isomerization of lactose to allolactose. Now the second structural gene is called Y gene, which is about 1254 base pairs. This is involved in synthesis of enzyme called lactose permease, which is found in cytoplasmic membrane, and it helps in transport of lactose into the cell. So it allows lactose into the cell. That's why it is named as permease enzyme, lactose permease. Now the third structural gene is called A gene. A gene will synthesize the enzyme called trans-acetylase and adds acetyl coenzyme A to beta galactoside. So its function is not very specific. It is not directly involved in any lactose metabolism. So it is not a very important function this gene will perform. Now next, operator gene. What is the operator gene? First position lies upstream to structural gene. Now what is the position? What is the upstream position? It is towards the 5' end of coding strand. So here we can say operator gene, these are structural gene upstream towards this end. If this is a 5' end and this side if it is a 3' end, then it is towards the upstream. Operator gene upstream to structural gene. All towards this side they are upstream, the other side they are downstream. So operator gene is present upstream to structural gene, and if it is on, then transcription will occur. If operator gene is on, functional, that means if it is free, then transcription will take place. If it is off, then structural genes, they cannot function. It gets attached to repressor protein. Repressor protein, which is formed from the inhibitor gene or repressor gene, that will bind to operator. So the mechanism we will see. Next is the promoter gene. This lies upstream to operator. We have just seen upstream to operator, the promoter will lie. So positions you should be very, very clear, children. You should know the position exactly. So if we take, there is a promoter here, then there is an inhibitor or regulatory gene, another promoter, then operator, and then there is a Z, Y and A. This is say if it is one strand 3' to 5', this is another strand 5' to 3'. You know this 5' to 3', this is called as a coding strand, and this is called as a template strand. So if we take operator, it is present towards 5' to structural gene. If we take promoter, it is present upstream to structural genes or to the operator gene. So what is the position given for promoter? Is lies upstream to operator. And its function is RNA polymerase will bind to promoter and then moves along operator and structural genes. So it binds there. If it is, this is the promoter, the RNA polymerase will bind at this place and then it will move along, finally transcribing the structural genes. So promoter, near the promoter region, it will first identify the promoter and then starts the process, then start initiating the process. And finally, structural genes can produce the respective protein. That is the function of a promoter gene. Now, let us see the next one. Regulatory or repressor gene or inhibitory gene. So, its position, as we have just seen, how do we read the position of a regulatory gene, or this is the inhibitor or regulatory gene? It is present upstream to the operator. Upstream to the operator or structural. And this gene, this inhibitory gene or regulatory gene will synthesize repressor protein that binds to operator and stops transcription. When repressor protein binds to operator, then it will stop the process of transcription. It is an allosteric molecule and changes its activity when another molecule is present. Another molecule. So, this repressor protein, which is an allosteric molecule, if any other substance is present, repressor protein will bind to that substance, then it cannot bind to the operator. And it is a constitutive gene. This gene is a constitutive gene. That means regulatory gene will always produce a repressor protein. It always produces. So it is called always functional. So it is called as a constitutive gene. Now next let us see what is inducer. Inducer here is allolactose. Inducer means in its presence transcription will take place. That is called as inducer. Inducer is allolactose, which is an isomer of lactose and functions as inducer. So when lactose is present, lactose is first converted into allolactose with the help of enzyme beta galactosidase. And then this allolactose will function as inducer. So what is the function of inducer? Inducer will bind to the repressor and then changes its conformation. And now repressor cannot bind to operator. Then when it cannot bind to operator, then RNA polymerase will help the expression of structural gene. So this is in general in the diagram we can see. So this is the inhibitor or regulatory gene. This will always produce first mRNA by transcription. Then it produces a repressor protein. This repressor protein, when it binds to operator, then RNA polymerase cannot bind to operator and promoter. So transcription cannot take place. So structural genes will be switched off. This will take place if inducer is absent. This type of mechanism we can see when inducer is absent. What is the inducer we have seen? Inducer is allolactose. That means if lactose is absent, lactose will be converted into allolactose. Suppose if lactose is absent, then the repressor protein, which is always formed from this gene, will bind to operator and then structural genes cannot function. So when structural genes cannot function, they will not be able to produce any enzyme. Now, when inducer is present, then what will happen? If inducer is present, then the repressor protein will bind to inducer. Inducer means it is allolactose. It will bind to inducer and then it becomes inactive. Inactive repressor is formed. So it can no more bind to operator. When it cannot bind to operator, then RNA polymerase will bind to promoter. It will first identify promoter. Then after binding to operator, it will start moving through the structural genes. And then it will transcribe the structural genes. So after undergoing transcription, this will produce mRNA called Lac mRNA. Lac mRNA will undergo translation. And after undergoing translation, it will produce 3 enzymes. This function we have already done. Beta galactosidase, permease and transacetylase. So these 3 enzymes will be produced. This is how the inducer will control the repressor. So repressor molecule which is always formed here is controlled by inducer, in absence of inducer and in presence of inducer. So in presence of inducer, repressor molecule will become inactive. So when it becomes inactive, transcription will take place. Active repressor is in the absence of inducer. So here repressor is controlling actually the process of, or it is regulating the Lac Operon. So a very low level of expression of Lac Operon has to be always present in this cell. Whether inducer is present or not, a very low level of expression is always present in the cell. Otherwise, there will not be any permease produced. If there is no permease, lactose cannot enter into the cell. Why this low level of expression always is required? Lactose, for entry of lactose. How lactose is entering? Due to formation of the enzyme permease. So if permease is not formed, lactose cannot enter. So a very low level of expression always is required. And this type of regulation in Lac Operon, this is called as inducible operon. If it is controlled by repressors, this type is called as a negative control also. It is inducible operon because there is an inducer present. And it is controlled by repressor, so it is called as a negative control. So Lac Operon with the help of repressor proteins and inducer, it is called as a negative control. There is also a positive control. Of course, it is not there within the syllabus and beyond, much beyond the level of 12th class. But for the sake of viewers, other viewers, and for the sake of the students who want to know what is positive control, because the word is mentioned in the textbook, I would like to discuss here. So let us see what is positive control of Lac Operon. Positive control means it is the glucose which is controlling, when it is controlling the regulation of Lac Operon, then it is called as positive control. So briefly, in an understandable way, I am going to give this. So it is due to high glucose concentration in cytoplasm. Suppose glucose is very high. A compound called cyclic AMP, this is called as CAMP, means it is called as a cyclic AMP. AMP, you know, it is adenosine monophosphate. So this cyclic AMP will be very low if glucose is high. Or in other words, if glucose is low, cyclic AMP is high. So when cyclic AMP is high, it forms a complex with another protein called as catabolite activator protein. So cyclic AMP must be maintained high for Lac Operon to operate. That is the most important point here. Cyclic AMP must be maintained high. So a high cyclic AMP is required for maintaining the Lac Operon, or for Lac Operon to function. So first important sentence I will write here. What is the important point which you should understand? Cyclic AMP is required in high concentration. Cyclic AMP is required in high concentration for functioning of Lac Operon. So for functioning of Lac Operon, high cyclic AMP is required. When is high cyclic AMP seen? When glucose is low. So what happens? When cyclic AMP is available, this cyclic AMP will form a complex with a protein called catabolite activator protein. This type of protein is synthesized for functioning of Lac Operon. It is synthesized in the cells, in the bacterial cells. It is called catabolite activator protein, and this protein will form a complex. So formation of this complex is very important. Catabolite activator protein and cyclic AMP complex. This is required for activation of Lac Operon, and this complex can be formed only if there is a high cyclic AMP. When is high cyclic AMP? We find more concentration when glucose is low. Suppose glucose concentration is increasing, cyclic AMP becomes very low, then Lac Operon cannot function. And this type of control by glucose, by presence of glucose, this is called as a positive control. So when glucose can become high? We know due to in Lac Operon, lactose is converted into glucose and galactose. By galactose metabolism breakdown, galactose can be converted into glucose also. Both are again isomers. Galactose can also be converted into glucose. Thus, glucose concentration can increase. So, when glucose is increasing, it is called as a positive control. Positive control. So, when glucose is increasing, that means Lac Operon cannot function or it becomes inactive. It slows down. That is how you have to understand the positive control. And this type of control is also called catabolite repression. It is called catabolite repression using this complex. When a theory is explained using this complex, that is catabolite activator protein and CAMP complex, cyclic AMP complex. And this catabolite repression actually is due to mutation of a gene which can synthesize this protein, catabolite activator protein. So this type of control is called as a positive control. You may get a question if you get slightly at a higher level. What is catabolite repression? Suppose you have to answer an objective question. What is catabolite repression? So simply you can understand it as it is the positive control of Lac Operon. That is first thing. I'll write down the point, 1 or 2 points, how you can answer any question if you get a as catabolite repression. First thing, first point we can understand it as it is a positive control of Lac Operon. Next point we can take it is seen at high glucose concentration. It is seen at high glucose concentration. So what happens at high glucose concentration? Cyclic AMP becomes low, and the complex, that is CAP, catabolite activator protein, and cyclic AMP complex is not formed. Then Lac Operon, if this complex is not formed, then Lac Operon is non-functional. This complex is required for functioning of Lac Operon. So Lac Operon is not functional. These are the points which you have to remember under catabolite repression. So I'll read the points once again for your understanding. It is the positive control of Lac Operon. It operates at high glucose concentration. And if there is a high glucose concentration, this compound cyclic AMP is low. If cyclic AMP is low, then this complex catabolite activator protein and cyclic AMP complex is not formed. If the complex is not formed, then Lac Operon is non-functional. So these points, to understand what is catabolite repression and positive control, you have to understand. I hope I am clear, my dear viewers and my dear students. So here we can conclude the topic. we completed what is Lac Operon. In my next session, I'll be discussing about tryptophan operon also. Tryptophan operon in my next video. So I hope you have understood, and those who have joined my video, joined my session for the first time, as usual, you please subscribe my channel so that whatever videos I upload, you will be able to get the notifications. For notifications, you have to also press the bell symbol so that as and when immediately when I upload a video, you will be getting the notifications. And I always welcome your suggestions also and your doubts. Doubts you can express in the comment section. I'll be going through the comments. If I find any clarification, any doubts, I will definitely rectify or clarify in my next video session. So thank you very much, children, and all my viewers, all my subscribers. I thank all my subscribers. The intention is for me is to reach maximum number of viewers so that I can help all youngsters, those who come into this field, teaching field, and also my dear students. So let us stop here. We keep talking in every video, and we can have a general talk also. In my next video, mostly I'll be giving a face-to-face video. This I have given because there are many points to give, and I felt like a notes if I prepare and then present it to you. This will be very useful. The tryptophan operon is a very small one, just a diagram and little explanation so that I can come front of you in front of camera and then explain it to you. So thank you for listening, children, and all my viewers. Thank you very much. Have a good day.